Welcome everyone to the Advanced Science Virtual Oncology Conference. This is the Biomarker Discovery track. My name is Alessandra Cesano, and I have the privilege of moderating today's session, which include 5 exciting talks focusing on novel approaches to biomarker discovery in the field of tumor-host interaction. Before starting, I have a few housekeeping notes. For a complete biography on today's speaker, please visit the tab at the top of your screen. And also, if you have any questions, we encourage you to submit them in a Q&A box. Our speaker will respond back to you via email. Before starting with the presentation, I just have 2 slides to briefly frame the context of today's presentation. The tumor microenvironment is a multifaceted ecosystem characterized by profound cellular heterogeneity, dynamicity, plasticity, and a series of complex intracellular crosstalks. Schematically, we can recognize different layers of heterogeneity in the tumor microenvironment. A layer that include, as a first one, the cancer cell genetic feature, oncogenic pathway, driving mutation. A second layer that include non-malignant cells, like the immune cells or the fibroblast. And a third layer that involve cellular crosstalk, which is affected by expressed and secreted molecules, as well as carried antigens and receptor diversity. The striking responses obtained with immune checkpoint blockade, for instance, have demonstrating the enormous potential of anti-cancer treatment that target the tumor microenvironment components instead of tumor cells. Therefore, a holistic, multiplex, and multi-omic, an integrated approach in the investigation of the different facets of the tumor microenvironment is needed to master its modulation and achieve an effective control of tumor growth. So how we can do that? Well, this slide summarizes the main approaches for multi-omic profiling of the tumor microenvironment by schematically dividing them based on the level of resolution, particularly full tumor sample resolution, or cellular single-cell resolution, or spatial resolution. Each level of resolution has different characteristics which can be used to optimally address specific biological. The bulk profiling, the full tumor sample profiling, is the most standardized and widely used and provides high throughput representation of molecular features of the tumor microenvironment as a whole. It has been and still is the main horsepower in the host-tumor interaction profiling and is at the basis of some of the main discovery in the biomarkers. Unlike the average representation that is provided by the bulk approach, emerging single-cell technology allow for the generation of omic profiling at the level of the single cells. This allows for the dissection of the intratumor heterogeneity and to shed light on rare cells that can play a role in cancer progression or specific cell subset, for instance, like immune cell subset. However, currently the cost and the technical complexity, particularly on the side of the computational side of things, limit the throughput in terms of both the number of features that can be examined as well as the number of cells that can be assayed at a single time. Finally, another emerging technology are the imaging technology that allowed for omic data set to be generated from tumor tissues slide that retain the information relating to the spatial resolution, thus providing an additional layer of information for a better understanding of the tissue biology. And on this latter approach, we will hear from many of our speakers today. So with this short introduction now, let's move to our first talk. It is my pleasure to introduce you to Dr. Aubrey Thompson, Professor of Cancer Biology, Co-Director of the Breast Cancer Translational Genomics Initiative Mayo Clinic Comprehensive Cancer Center, Jacksonville, Florida. The title of his talk is Spatially Resolved Evaluation of Triple-Negative Breast Cancer, Expand Knowledge of Immune Cell-Tumor Cell Interaction. Please join me in welcoming Dr. Thompson. I will now turn the presentation over to you.
Good afternoon. It's a pleasure to be with you this afternoon to be able to share with you some highlights of the work that we've been doing using the NanoString Genomics digital spatial profiling instrument to begin to map the immune landscape of triple-negative breast cancer. Before I begin, I have to emphasize one thing. This is very much a collaborative project, and everyone listed here has been a significant contributor to this project, but a couple of people really deserve special mention. Jennifer Kachurgas in my laboratory has probably run more DSP samples than anyone outside of Mayo Clinic, and she's, outside of NanoString. And she has really been the workhorse in all of the data that I'm going to show you. In addition, I need to cite Doug Heinerfeld. Doug has been involved in these experiments from the very beginning, was involved in experimental design, in data collection, data interpretation. And he has been a stalwart contributor throughout all of these studies. So what I'm going to talk to you a little bit about today initially is some studies that we've done with a set of triple-negative breast cancer that came from the FinXX adjuvant trial of fluoropyrimidines. We've done a number of things with these studies, but what I'm going to talk to you today is about data that we've obtained using a beta version of the Genomics DSP platform to begin to map the immune landscape of these samples. So from the FinXX trial, we selected two cohorts of 22 patients each. One of these cohorts had a median recurrence of 2.3 years. The other had a minimum of 9 years follow-up recurrence-free survival with 10.7 years median recurrence-free survival. So we're comparing two groups of tumors, one of which recurred and one of which did not recur. And these tumors are otherwise balanced with respect to their clinical and pathological features. We stained these tumors, these slides, with a cocktail of antibodies that contain oligonucleotide-targeted antibodies against 42 target reagents that was available to us. In addition, we used fluorophore-tagged antibodies against CD45, CD68, and pan-cytokeratin as morphological markers. So what we see when we stain the tumors is shown here. Pan-cytokeratin stains the tumor cells, tells us where the tumor cells are, and it's shown up here in green. Cyto13 is a nuclear dye, and so we use it to identify the cells that are within the stroma. We stained the tumors with CD68 so we could identify regions that were enriched for macrophages, and with CD45 to identify regions that are enriched for leukocytes. Within these tumors, then, we select a 300-micron region of interest, in this case, the interface between the green cytokeratin-positive tumor and the stroma. The stroma, in this case, is defined by the presence of nuclei. So the stroma really here, the important part of the stroma here is that it's blue, not that it's red. It appears that it's the red that we're selecting, but in fact, it's the blue. So we segment these. We segment them, each of these regions of interest, into two regions, the pan-cytokeratin positive tumor region and the cyto13 nuclear stain cytokeratin negative stromal regions. We selected three different kinds of ROIs from these tumors. We selected ROIs that contained little or no tumor-enriched ROIs. We selected ROIs that were enriched for CD45, and we selected regions of interest that were enriched for CD68. And all three of these regions of interest were then segmented into, for example, CD45-enriched tumor, CD45-enriched stroma, more specifically, CD68-enriched stroma and tumor that's adjacent to CD68-enriched tumor. And we collected these segments and then analyzed the number of antibody molecules that were bound to each of these discrete segments. The data are sort of summarized here. This is probably impossible to really make much sense out of, but the thing that I'll call your attention to is the red. The red indicates significant overexpression in patients that did not recur. And I think that what you can see here rather easily is that there's a lot going on in the tumor. The tumor is the tumor segments, the CD45 tumor, CD68 tumor, and the tumor without adjacent stroma have a large number of proteins that are differentially expressed. Whereas in the stroma, it's really rather quiet. The only things that show up are in all of the segments are GCMB and PDL2. So there's something going on in the stroma, in the tumor, excuse me. There's information in the tumor that is not recapitulated in the stroma. And for example, I'll show you here CD56, the NK cell marker. CD56 in tumor segments is associated with durable recurrence-free survival, in this case in CD60A in tumor that's adjacent to CD68 enrichment. So it's significant, CD56 is a significant prognostic indicator in the tumor segments, but is not a significant prognostic indicator in the adjacent stromal segments. So there's a lot of information that we're getting from the tumor that we're not getting from the stroma. We're going to focus in the rest of this talk actually on three particular proteins, two antigen-presenting cell proteins, CD40 and HLA-DR, and then the NK cell marker CD56. And as you can see in the table, all three of these are significantly associated with outcome in the tumor segments, but none of them are significantly associated with outcome in the stromal segments. So this is, again, consistent with the idea that there's information that we're getting in the tumor that's not being obtained if we simply look at the stromal segments. Now, the issue that comes up when you look at these data is, well, CD40, CD56, HLA-DR, those are fairly well-known lymphoid cell markers. What's the chance that when we see these things that are associated with outcome that it's a surrogate view of tumor-infiltrating lymphocytes or TILs? So is there really more information here than we would get if we had simply measured tumor-infiltrating lymphocytes? Well, we don't have classical H&E stained tumor-infiltrating lymphocyte data from the FinXX trial, but what we do have is very high-resolution digital images from tumors that are stained with CD45 and CD68. And my colleague Jody Carter developed a series of analyses using halo instrumentation to measure the number of CD45 positive, CD68 minus cells, to measure the density of these in each of the tumors. So this is essentially digital TILs analysis. So when we look at that, we see initially that tumor-infiltrating lymphocytes are in fact prognostic in triple-negative breast cancer. They should be. That's been known for a long time. But now the question becomes, is there anything other than this going on? And in fact, what we see is that tumor-infiltrating lymphocytes convey a certain amount of information, but not the whole story. So what I've presented here is CD45 positive cells plotted against the concentration of HLA-DR. Each of those dots represents a single tumor. The red dots represent tumors that recurred. The green dots represent tumors that did not recur, okay? Now, if you look at the lower left-hand corner of that slide, what you'll see is that tumors that have very low level of CD45 positive cells and low levels of HLA-DR, that those patients do very well. Those are mostly red. Those patients recur, okay? But if you look above that, if you look at the low TIL, high HLA-DR, what you'll see is that there is a population of tumors, that have low level of tumor-infiltrating lymphocytes, but have in fact high levels of TILs, high levels of HLA-DR, and those patients do perfectly well, okay? So in general, when we look at these data, what we tend to see is that HLA-DR in the tumor segments is a more robust prognostic indicator than total tumor-infiltrating lymphocytes. We looked at this in somewhat more detail in 3 dimensions here using CD40, HLA-DR, and CD56 to distribute the tumors. Again, each dot is a single tumor. Green is no recurrence, red is recurrence. These are stromal segments. So we've looked at the expression of these three markers in stromal segments. And as you can see, there's a considerable amount of heterogeneity within the stroma. But for the most part, the tumor samples that recur pretty much overlap with the tumor samples that do not recur. We see a very different situation when we look in the tumor segments themselves. What we see here is that the tumors that recur, the red dots here, cluster very tightly with low-level abundance of CD40, HLA-DR, and CD56. So the tumors that recur are uniformly cold for these three markers in the tumor segments. Conversely, the tumors that did not recur exhibit a great deal of heterogeneity. So they're fairly well spread out based on these three individual markers and distribute very differently from the tumors that recur. If we look at this in 2 dimensions, we see something even more striking. In this case, I've plotted HLA-DR expression against CD56 expression. And what you can see is that HLA-DR and CD56 are basically expressed in different subsets of tumors. So tumors that have high-level HLA-DR tend to have very low-level CD56. Tumors that have high-level CD56 tend to have very low-level HLA-DR. So what we're really looking at here is 3 different immune landscapes. We've got an immune landscape that contains very low-level CD56 and very low-level HLA-DR, and by and large, those patients don't do very well at all. We have an additional immune landscape that contains high-level CD56 and low-level HLA-DR. Those tumors, those individual tumors, tend not to recur. Likewise, tumors that have high-level HLA-DR and low-level CD56 tend to recur. So to summarize what I've shown you up to this point, I think that our data strongly indicate that the immune cells that are in intimate contact with the tumor cells may in fact be more informative than just looking at total tumor cells, as in stromal tumor-infiltrating lymphocytes. I think that our data indicate that tumors that have high-level expression of natural killer cell markers or antigen-presenting cell markers have better prognosis, and that seems to be irrespective of the presence or relative lack of tumor-infiltrating lymphocytes in those tumors. So we're seeing something that's really quite different from what we see if we simply measure stromal tumor-infiltrating lymphocytes. And then finally, as I emphasized previously, we're looking at 3 potentially distinct immune landscapes identified by the markers that I've shown you here. So we have a cold immune landscape. Those patients tend to do poorly. We have a CD56 or natural killer cell enriched, presumed to be a natural killer cell enriched immune landscape, and those patients do well. And then we have an additional immune landscape that's characterized by high HLA-DR. I could show you the same thing with CD40. They're both markers for antigen-presenting cells, and tumors that have high-level expression of either or both of those APC cell markers tend not to recur. So there's multiple immune landscapes that are detectable within tumor segments that are associated with favorable prognosis in triple-negative breast cancer. None of those are detectable within the stromal segments. I'd like to finish this afternoon by telling you some data from a preliminary analysis of a triple-negative breast cancer tissue microarray. This microarray contains 287 patients and was constructed by the Mayo Clinic Breast Cancer Spore, and we obtained it through the good offices of Matt Getz. This tissue microarray is really quite different from the FinXX samples that we've been discussing. So the FinXX samples were selected for early stage tumors, whereas the tissue microarray was essentially drawn from all Mayo Clinic triple-negative breast cancer patients. So there's no selection for tumor stage or grade. In fact, there's no selection for therapy among these patients, as opposed to the FinXX trial in which all the patients were treated with the same therapy. So this is a tissue microarray. It was constructed to have 2 cores from each tumor. So we've got a maximum of 4 segments from each tumor, 2 from each core, as opposed to 24 segments that we had with the FinXX tumor. So there's considerably more statistical assurance in the quality of the data from the triple-negative breast cancer from the FinXX trial than it is from the TMA. The cores were selected for the TMA by a pathologist who simply went in and selected for cores that had high tumor density. So this is really quite different from the selection process that was used from the FinXX trial. And these cores are actually fairly similar to the tumor without adjacent stroma regions of interest that we selected from FinXX. And then finally, the FinXX analyses were done on a beta version of the genomics DSP machine using beta reagents. The TMA was analyzed on the commercial version of the machine using commercial reagents, which involves some subtle changes in the antibodies that were used to measure these proteins. So what I'm really telling you is that there's almost nothing in common between the FinXX samples and the tissue microarray samples, other than the fact that they're both triple negative. So it's really rather surprising when you look at the data. This is a plot of the 39 proteins that were common to both the FinXX and the tissue microarray analysis. Each spot there, each dark spot, black spot, is a single protein. And you can see that there's actually a remarkable correlation between the normalized counts for every protein in the FinXX samples genomics DSP technology, the ability to compare two very, very different sets of tumors at the level of normalized protein expression and get this kind of correlation is almost breathtaking. At any rate, we went on and we carried out a series of experiments. We measured the intensity, we measured the abundance of the proteins using DSP technology. And as you can see here, that we got for the tumors that recurred in the tissue microarray were very similar to those that we observed with the FinXX. The tumors that recurred cluster very tightly with respect to these three markers. These tumors are cold, whereas we look at the tumors that did not recur, we see essentially the same thing. We see a great deal of heterogeneity within the tumors that did not recur, and that heterogeneity again reflected by differences in the expression of these three markers, CD40, HLA-DR, and CD56. When we look at this again in 2 dimensions, you see something that's really quite similar to what we saw with the FinXX data. We're seeing 3 different, or a minimum of 3 different immune landscapes. There's a high HLA-DR landscape, a high CD56 landscape, and then a cold landscape. And those landscapes really differ with respect to their prognostic significance. The cold tumors tend to recur, whereas the high HLA-DR, low CD56 do not, and the high CD56 likewise tend to have better prognosis. So to wrap this up, a few things I think warrant being said by way of conclusion. I think that the data show you in this brief presentation, and again, I emphasize that what I've shown you are mostly highlights from these studies, but I think that what the data indicate is that the genomics DSP platform is really providing a powerful new way for us to begin to look at the immune landscape of breast cancers. In fact, not only breast cancers, but other sorts of tumors as well. I think the most interesting thing that we're beginning to see is that there is information within the tumor compartments that's going to give us new insight into immune features that are associated with therapeutic outcome in breast cancer. I think that we're seeing that there are multiple immune landscapes. I've shown you, I believe, evidence for 3 immune landscapes, but undoubtedly there are other types of immune landscapes within the triple-negative breast cancer. These immune landscapes appear to be mutually exclusive. That is, there are some tumors that have high CD56 and they do very well. There are other tumors that have high HLA-DR and they also do well, but those are not the same tumors. Different immune landscapes associated with outcome. And then finally, I think the data overall indicate the fact that analysis of protein abundance in a spatially defined manner is going to really add a whole new dimension to biomarker discovery, and to enable us to begin to ask fundamental questions about the immunobiology of breast cancer and the relationship between immunobiology and therapeutic outcome, in this case in terms of chemotherapy, but also in terms of immunotherapy as well. So I'll finish up by expressing my thanks to the patients who contributed their samples to the FinXX trial, to Heike Janshu, who was the principal investigator on that trial. Without him, we obviously could not have gotten these samples. Torsten Nielsen, the University of British Columbia, actually initiated these studies, which were funded in part by the National Cancer Institute, the Breast Cancer Research Foundation, and Donna Deegan. And then finally, I need to express my profound thanks demonstrating not only Doug Hinterfeld and Sarah Warren and Heather Ann Brower, who were involved in experimental design and interpretation, but specifically to the outstanding genomic support team who held our hands through all of these now almost 600 tumors that we've analyzed to date. So I'll stop at that point. Thank you for your attention, and I'll attempt to answer any questions that you may have.
Thank you, Dr. Thompson, for a very informative presentation. I have a few questions for you. Your data seem to imply the Stroma tumor-infiltrating lymphocytes may be relatively unimportant to the outcome of triple-negative breast cancer. Do you agree with this interpretation of your data?
No, I don't agree with that at all. I did not mean to give you that impression. Certainly, we know that stromal tumor infiltrating lymphocytes are of prognostic significance in triple-negative breast cancer. And there's a lot of discussion about whether or not evaluation of stromal TILs should become part of routine clinical practice. So I think that that's been a very useful way to look at the immunobiology of triple-negative breast cancer. But I think the reality of it is that there's a great deal more information that we can have above and beyond just simply looking at stromal TILs. We've looked at stromal tumor infiltrating lymphocytes because that's what we could do, because we could stain the stroma, we could count the lymphocytes. It's very difficult to count lymphocytes in tumor by conventional H&E. It's very difficult to count macrophages. But the DSP technology now gives us the ability to look at protein markers that are associated with natural killer cells or with APC cells, or macrophages, and to look at that within both the stroma and the tumor. So no, I'm not saying that stromal TILs are not important. They certainly are important. I'm just saying that the immune cells that ultimately I think are going to be more important are the immune cells that are within the tumor. It's the immune cells that are actively engaged with the tumor cells, rather than the immune cells that are out in the stroma, kind of wondering what they're supposed to do next.
Thank you. Along the same lines, do you have any insights into the mechanisms that underline the difference with respect to heterogeneity that you observe in tumor segment from patients who recur and those who do not?
That's almost a version of the question that I just answered. We do not at this time understand why some tumors have high levels of CD56, other tumors have high levels of HLA-DR or CD40. Obviously, that's a very important fundamental biology question, but we have not yet been able to tease out the answer.
Thank you very much. Interestingly, your data identify a subpopulation of tumors that are immunologically cold but do not recur. Have you looked at this cold tumor to see if there are any other immunological features that might be prognostic?
So the question of validation comes up when you're looking at any new technology of this sort. One attempt that we've made at validation, obviously, is to do immunohistochemistry using antibodies against some of these markers. The problem that we've run into in trying to do that is that immunohistochemistry is actually relatively insensitive, and many of these markers that we're looking at are not high abundance markers. DSP is exquisitely sensitive, and so we can measure things, we can quantify things, and quantify changes in immune proteins using DSP at a level that's far, far below the lower limits of detection of immunohistochemistry. So that method of validation has not been successful for us thus far. I will say the following thing. I think that the data that I presented in comparing the FinXX tumors to the tissue microarray tumors does speak to one level of validation. The observation that with those two radically different tumor subsets, that when we look at the abundance of a large number of individual proteins in those two subsets and show that they correlate very highly from one to the other, I think that does speak to the robustness of the technology. It doesn't necessarily mean that the technology is accurate, but it does mean that it's reproducible. So I think that at this point, validation is a question that we're all concerned with, and we and a number of other laboratories are working on that at this time.
Thank you very much. Finally, have you attempted to validate any of your observations using orthogonal technologies?
Not yet. Obviously there is a population of triple-negative breast cancer patients whose tumors are cold. Most of those tumors recur, but some of those cold tumors do not recur. And so presumably there are features that we can discern within that subpopulation of cold tumors that do not recur. Presumably that may be another immune landscape. Alternatively, it may have nothing whatsoever to do with the immune system, but we're obviously aware of that potential question, and it's a subject of ongoing investigation.
Okay.
So in summary, I think that we're entering a new field, the field of spatial biology, and that we can look forward to fundamental new observations on the nature of breast cancer, the way that breast tumor cells interact with immune cells, and the consequences of those interactions with respect to therapy, ultimately, I think, contributing to the overall goal that we're all working for, and that is to reduce breast cancer mortality. Thank you for your attention.
Thank you again, Dr. Thompson, for your presentation and discussion.